Contractions of a whole muscle show variations in the development of tension. For example, the same muscles that can lift a potato chip can also lift a six-pack of soda.
A whole muscle does not respond to stimulation in an all-or-none fashion. Instead, it shows variations in the development of tension or force of contraction.
In this topic, we will examine three factors which affect muscle tension. 1. Frequency of stimulation. 2. Number of motor units recruited. And 3. Degree of muscle stretch.
We will begin our study of whole muscle contraction by looking at how a muscle responds to a single stimulus by making an isometric contraction.
A muscle contraction in response to a single stimulus of adequate strength is called a muscle twitch. Click the stimulator button to elicit a muscle twitch. The graph shows a tracing of the muscle contraction. The short vertical line marks the time when the stimulus was applied to the muscle. If you would like to see this animation again, click the stimulator button. When a second stimulus of the same intensity is applied to a muscle before the completion of the relaxation period, a second contraction occurs, adding its effect to the contraction resulting from the first stimulus. This is known as temporal summation or wave summation and results in increased muscle tension. The stimulator has been set to give 2 stimuli to this muscle. Click once on the stimulator button to see the response to a double stimulation. The resulting graph shows several aspects of the mechanical activity of muscles when the frequency of stimulation is altered.
The phases of this graph include trepa, temporal summation, incomplete tetanus, complete tetanus, and fatigue. Click on the label for each region of the graph for more information. The first few contractions demonstrate trepa, or the staircase effect. Look closely to see that even though the strength of contraction increased, relaxation was complete. This shows that temporal summation was not responsible for this effect. The increase in tension may result from increased muscle warming and efficiency of enzymes. Click the stimulator to see muscle activity during trepa, then click another region of the graph.
With increased frequency of stimulation, the muscle exhibits shorter contraction-relaxation cycles. This condition is called incomplete or unfused tetanus. Note that some degree of relaxation is visible after each contraction. Click the stimulator to see muscle activity during incomplete tetanus. Then click another region of the graph. With rapid multiple stimulation, the contractions fuse into a smooth, continuous total contraction without evidence of any cyclical relaxation. The muscle is now in a state of complete or fused tetanus. Abundant intracellular calcium provides continual availability of binding sites on actin for cross-bridge cycling. Click the stimulator to see muscle activity during complete tetanus. Then, click another region of the graph. With continued rapid stimulation, fatigue becomes apparent as the muscle is no longer able to sustain its level of tension, but gradually elongates. Fatigue is due to multiple factors: buildup of acidic compounds which affect protein functioning, a relative but not total lack of ATP, and ionic imbalances resulting from membrane activities. With rest and adequate blood supply, fatigue conditions are corrected, and the muscle will again be ready to respond to stimulation.
Click the stimulator to see muscle activity during fatigue. Then click another region of the graph. Or if you have seen all parts of the graph, click the Next button to proceed.
After the first 5 stimuli, the frequency was increased, which caused temporal summation. Notice that this resulted in a continual increase in tension as one contraction was added to the previous contraction. The increase in tension may result from increased availability of intracellular calcium. Click the stimulator to see muscle activity during temporal summation. Then click another region of the graph.